We're going to go ahead and move into our next panel, and I am going to bring to the forefront Dr. Felix Nitschke. Dr. Nitschke is an assistant professor in the Department of Pediatrics and Biochemistry at UT Southwestern Medical Center. Dr. Nitschke's research is focused on understanding the principles of functional and pathological glycogen metabolism and on establishing a novel enzyme replacement platform against multiple rare diseases. In 2021, Dr. Nitschke was awarded a $1 million bike ride research grant to support the ongoing development of a novel therapeutic strategy that removes polyglucosan bodies. He serves as co-chair of the Foundation's Scientific and Medical Advisory Board. Thank you so much for being here, Dr. Nitschke.
And thank you so much for the introduction. And I think the next session is going to be awesome. We're going to hear from a big lineup of researchers that I kind of admire and look up to. One of them is Dr. Matthew Gentry from the University of Florida. I'm going to introduce him as the chair in the Department of Biochemistry and Molecular Biology in that university. And he's going to talk about protein therapy in APBD. Then there's going to be Dr. Burj Menassian, Division Chief of Pediatric Neurology in the UT Southwestern Medical Center. And then we have Dr. Soo-Jen Choi. She is Research Associate at Duke University, talking about APBD gene therapy. And then we have Dr. Wyatt Yu, Professor of Structural Biology at Newcastle University. And he's going to talk about small molecules. So we have protein therapy, antisense technology by Dr. Menassian, gene therapy, and small molecules, all treatments in development. And I'm really kind of looking forward to seeing those 4 presentations. And as in the last session, we're going to have basically questions answered, Q&A in the end of those 4 talks, after all 4 talks have finished. And please use the chat or the Q&A function to enter your questions. And then I would just give it over to Dr. Menassian. Please take it away and tell us about ASOs.
Thank you. Thank you, Felix and the organizers. It's really nice to be part of this community. We used to meet in New York and eat some very nice food together. Now in the day of Zoom, we only see each other in little rectangles, but it's great to be here nonetheless. So like Matt said, this theme of single drug for multiple diseases will apply in this one here that I'm going to talk about. Briefly, as you know, APBD is caused by a deficiency of branching enzyme. So on this picture on the left side, you have the way normal glycogen looks like. And as you see, it is very much branched. And on the right side is how polyglucosans look like, the ones that form the polyglucosan bodies of adult polyglucosan body disease. These polyglucosan bodies drive a neuroinflammation and a neurodegeneration, which we strongly believe underlies the main aspect of the disease, the neurodegenerative aspect of the disease. So to understand what's going on, one should review glycogen metabolism briefly. And the relevant part is quite simple to understand, actually. So you have 2 enzymes that must work in tandem together. Glycogen synthase is the enzyme that elongates chains. So each of these circles is a glucose molecule. So glycogen synthase adds one glucose after another, and so makes a chain of glucoses. Branching enzyme is the enzyme that makes the branch points. So you can already see that if glycogen synthase and branching enzyme are working in concert, meaning the glycogen synthase is not going any faster than the branching enzyme, or the branching enzyme is not going any slower than glycogen synthase, then you have this kind of rhythmical, gradual branching pattern, and you get a molecule that looks like a sphere, and this molecule is nicely soluble, no problem. But what we have in APBD is this situation. The APBD patients have decreased branching enzyme. So what happens in this case now, glycogen synthase is outpacing branching enzyme, and the branches that are being formed are too long. And when the branches are too long, the molecule is unstable and precipitates, meaning it kind of goes out of solution. And each time one of those molecules precipitates, they aggregate and accumulate over each other and form these polyglucosan bodies. Incidentally, this glycogen synthase enzyme is regulated by various ways, including by these laforan-malin proteins, which are the proteins involved in Lafora disease, which is the sister disease, if you like, for APBD. So laforan and malin keep glycogen synthase in check. Because again, we don't want glycogen synthase to outpace branching enzyme. Every time it does that, you get a polyglucosan that forms and you get a polyglucosan body eventually, and then the disease. So in APBD, we have reduced branching enzyme. You will hear later in this session how one could replace branching enzyme through gene therapy. And indeed, you will hear projects going on towards that. What I'm going to talk about today is a different approach. What if we reduce the activity of glycogen synthase to return the balance that we need? So if we are able to, because branching enzyme is decreased in APBD, if we're able to decrease glycogen synthase, then we return to our balance, and glycogen synthase is now no longer outpacing branching enzyme, and then we fix the problem. So to do this, we need basically ways to downregulate glycogen synthase activity. And you will hear later today, one way is to use a small molecule drug that acts on glycogen synthase and decreases its activity. What I'm going to talk about is an antisense oligonucleotide drug. This is a small piece of DNA or an oligonucleotide that can be given and that will target the RNA that makes glycogen synthase and will downregulate glycogen synthase. And so these experiments have been done. This is work by Dr. Nitschke, Sylvie Nitschke, not Felix Nitschke. This is a wonderful scientist who happens to be the spouse of Felix. And you can see in these slides how when you give these mice an antisense oligonucleotide targeting glycogen synthase, if you look where my arrow is here, the amount of polyglucosan bodies decreases dramatically. And the pictures here show it as well. Now, these experiments have been done in mice. The mice genome is different than the human genome. So in this case, an antisense oligonucleotide was used that works in the mouse, so a mouse ASO. So one needs to do the same experiments using a human ASO if we are eventually to develop the human ASO for use in human subjects. So again, Dr. Sylvia Nitschke and others have worked on developing a mouse which, instead of having the mouse glycogen synthase, has a human glycogen synthase. So the mouse glycogen synthase is removed and the human glycogen synthase is put in. We call this a humanized mouse. This mouse is a mouse, except that it has human glycogen synthase instead of mouse glycogen synthase. What this allows us to do is to test drugs that target the human gene or the human protein, or in this case, the human mRNA for glycogen synthase, to develop towards a treatment. This type of mouse we have already, as I mentioned, but we have it in the Lafora disease background, meaning the mice have Lafora disease and human glycogen synthase, and a lot of work has been done in that. And basically, we have a drug now called ION-283. Sorry, the number 3 is not visible here. ION-283, this was developed in collaboration with Ionis Pharmaceuticals and other people here, including Dr. Gentry, Dr. Nitschke, etc. And this drug is now FDA approved for clinical trial in Lafora disease. That will start before the end of this year. For the APBD, we now have the mouse with the humanized glycogen synthase in the APBD mouse. So we have APBD mouse, the original mice made by Dr. Ackman, that instead of having the mouse glycogen synthase, have the human glycogen synthase. And so these same experiments can be done in those mice, and this ion 283 potentially could become a treatment that could go into clinical trial for APBD. I'm just going to say a couple of words on the clinical aspects, because developing drugs is not just the work you do in mice. The clinical aspect is very, very important. And I think that's that's one of the main things holding treatments back in APBD. And the issue is that APBD is a relatively slowly progressing disease. Although it's a severe disease, it's slowly progressing. And so it's hard to do a clinical trial in something that progresses relatively slowly. What you're seeing here is from a childhood disease, which is very, very rapidly progressing. It's CLN2, a very severe neurodegenerative disease of children. And you see in this diagram how their motor and language scores dramatically decline over a period of a couple of years. And so this is so fast that in a 2-year window, you can do a clinical trial with whatever drug. In this case, the drug was a kind of enzyme, basically the enzyme that's mutated in this disease was replaced in the brains of these patients. And so on the right, you're seeing the clinical trial that was done, and you could see in a matter of weeks how the patients who usually crashed in the red are now not doing so. So it's easy to do a clinical trial when things are fast like this. Lafora disease is intermediate, and you see now we're talking about years instead of months and weeks. And if you just follow this blue line, you see the decline in these patients over a period of 2 years. So there is a reasonable window to do a 2-year clinical trial. The difficulty, the challenge in APBD is that the years are so long of the progression of the disease that it's harder to do a clinical trial with whatever drug. I'm bringing this up just for discussion, but I'm finishing here to say that, you know, we already have a drug for Lafora disease that, for reasons I explained, can be applied to APBD. The community needs to find a way to kind of leverage that and get that drug somehow into clinical trial in APBD or some other way into patients. But there are other drugs too. You've heard of small molecules from Dr. Kahlon, all the exciting enzymes that Dr. Gentry described that will also be effective and so on. But the APBD community needs to figure out how we're going to do clinical trials in APBD. Just put that out for discussion, and I'll just stop here.
Thank you, Bersh, for this very interesting and engaging presentation. And without much ado, I will just introduce again Soo-Jin Choi, Dr. Soo-Jin Choi from Duke University, and she's gonna speak about gene therapy in APBD.
Thank you so much. Thank you so much. My name is Soo-Jin Choi, and I'm honored to presenting our work here. Today, I'm talking about exploring the therapeutic benefits of AAV-mediated glycogen clearance in the CNS using a mouse model of glycogen storage disease 4. So first of all, I would like to briefly introduce the background of glycogen storage disease 4. So many folks already discussed about this, but glycogenesis is the synthesis of glycogen from glucose in the body. And here is the key enzyme involved: glycogenin, glycogen synthase, and glycogen bridging enzyme. Glycogenin initiates this process by self-glycosylation, and glycogen synthase elongation the glycogen chain, and glycogen bridging enzyme introduce the bridging point here. However, mutations in the GBE1 gene cause glycogen storage disease 4. characterized by the accumulation of insoluble polyglucosan bodies in the body. GSD4 present with hepatic, cardiac, and neuromuscular involvement. And also there is another form of glycogen storage disease we called APBD, result from the tyrosine point mutation in GBE1. Exhibit symptoms like spastic gait, progressive neurogenic bladder, and cognitive decline. For APBD research, we used knock-in GSD4 mouse model with tyrosine 329 serine mutation, successfully recapitulate APBD patient phenotype. Polyglucosan body accumulation was observed in liver, muscle, and brain and spinal cord. And here, as you can see, kyphosis and lectoprops appeared in both genders after 6 months. And this model exhibits APBD like symptoms, impaired motor function and gait difficulty, neurogenic bladder, and also altered sensory nerve conduction. Here is our behavior test data show significant motor function decline in GSD4 mouse compared to wild type between 3 and 6 months of age. This PSD staining figure illustrates the progressive accumulation of polyglucosan bodies over time in GSD4 mice. In 6 months old wild type mice fasted for 24 hours, no glycogen accumulation was observed in the brain and spinal cord. However, GSD4 mice displayed early polyglucosan body accumulation from 1 month of age, and between 3 and 6 months age, the polyglucosan body accumulation increased and persisting up to 12 months of age. And polyglucosan body predominantly accumulated in the brain's white matter, and particularly around the Purkinje cell layer and granular cell layer in the cerebellum. And the confirmation of this PSD staining data was obtained through glycogen contents assay. As you can see here, negligible glycogen accumulation in the wild type. However, increasing accumulation with age in GSD4 mice in brain and spinal cord. To optimize neuronal targeting in AAV gene therapy, we utilized synapsin-1 promoter to induce human GBE protein expression. And 6 weeks of age mice received AAV via tail vein injection at a dose of 2E14 pg per kilogram, and administered over varying treatment duration, 3, 6, and 10 months, and behavior tests were conducted before sacrifice them. We initially administered AAV treatment for 2 weeks in short-term study to verify successful induction of GBE protein expression. As you can see here, Western blot analysis validated GBE protein expression in the brain. And PSD staining data showed polyglucosan body accumulation in untreated GSD4 mice in 2 months old mice, the red arrow indicated. However, after AAV treatment, mice exhibit no glycogen accumulation in the brain. Now I will present the result from our long-term AAV treatment group. AAV was administered at 6 weeks and with treatment at 3 and 6 months. To evaluate AAV-induced glycogen clearance, we conduct various behavior tests prior to sacrifice. At 3 months, as you can see here, treadmill test showed similar performance among wild type, untreated, and treated GSD4 mice, despite significant polyglucosan body in the GSD4 mice. However, by 6 months, untreated GSD4 mice present poor performance than wild type. Following AAV treatment, GSD4 mice performed comparably to wild type. And this video was recorded 6 months post-treatment. In the inverted hang test, untreated GSD4 mice displayed significantly declined performance than wild type mice, but post-AAV treatment improvement were evident in GSD4 mice. These beneficial effects persisted for up to 6 months on treatment. Conversely, in beam walking test, as you can see here, at 3 months, there is no difference between 3 groups. However, at 6 months, untreated GSD4 mice present a struggle to cross the beam. I will show you the video. Like this. But while AAV treated GSD4 mice showed improvement comparable to wild type. In long-term cohorts, we verified significant expression of GBE protein by Western blot like this. And also immunostaining highlighted the predominant expression of AAV-induced human GBE in neuronal cells. Additionally, in the spinal cord, we utilized GFAP as an astrocyte marker. The expression of AAV-induced GBE did not localize with GFAP, meaning significant expression of AAV in neuronal cells. And this expression persisted consistently throughout the 6 months treatment period. We also examined astrogliosis in the GSD4 mouse model, illustrated in this figure, showing GFAP-stained astrocyte activation. At 6 months, wild type mice displayed minimal GFAP signal, whereas untreated GSD4 mice of same age exhibit increased astrogliosis in the hippocampus and anterior horn of spinal cord. And remarkably, AAV treatment resulted reduced astrocyte activation. Building on the previous PSD figure, we observed a significant accumulation of polyglucan body in the brain, 3-month-old mice, indicating the red arrow. However, 3 months after AAV treatment, PB accumulation in the brain was effectively prevented. However, after 6 months post-treatment, a significant accumulation of polyglucan body was detected via PSD staining, as you can see here. Is founded near the Purkinje cell layer or granular cell layer. Unfortunately, the therapeutic efficacy of AAV treatment diminished by 10 months post-treatment. We've done several behavior tests, but as you can see here, the AAV treated efficacy was diminished in this group. And also we saw the polyglucan body accumulation in the brain. However, by the Western blot, we also saw the GB expression was still there. So my time is out. So here is my summary. So in summary, with the AAV gene therapy, we saw the initial benefit of improving motor coordination and prevention of polyglucan body accumulation. However, it's not enough to cure this polyglucan body at all. So this highlighted the complexity of disease mechanism. Thank you so much for your attention.
Thank you, Dr. Choi. That was very informative and exciting, the work that you presented there. All right, so then we're already getting to the last speaker of this session, Dr. Wyatt Yu. Professor of Structural Biology at Newcastle University. Dr. Yu, please go about it.
Hi, everybody. Hi, Felix. I hope you can hear me. I'm just double checking. I'm on the road and I heard some feedback in my case, but if you are fine, that's great.
Sounds great, Dr. Yu.
Okay, that's good. Well, thank you so much for the kind invitation. And sorry I cannot join the other sections a lot today, but it's really great to be in this community as a basic scientist, and I'm very grateful for the support and the lessons learned in being part of this community. I think I'm very grateful to be following after the excellent speakers who have provided the background of APPD, but also have provided the rationale for targeting glycogen synthase today. Suffice it just to say that our approach is to think forward for small molecule therapies towards inhibiting glycogen synthase, as shown in part of the glycogen biosynthetic process that the other speakers have described. Yeah, we focus on small molecules. You might ask why. Of course, we understand some of the potential benefits of small molecules, yeah. But also practically, the type of work we do or the expertise we have in terms of understanding protein structures and 3D shapes are really quite attuned to the use for screening of the design of small molecules, which is what my focus in this work is. I thought of spending the next 5 minutes or so on providing a general background and perspective on how we do it. Yeah, and apologies for not showing much data, but I thought to have a perspective to think together, it might be more helpful. We're structural biologists, and because what that means is we're interested in shapes, and we're interested in using our knowledge of shapes to identify and screen small molecules. And 2 years ago, we really am grateful and delighted to be funded by this $1 million bike ride grant, and through all the generous support and hard work for this grant. We carried out we are setting up a screening platform called DNA Encoder Library Screening. Yeah, and what that means is that we immobilize, let's say, glycogen synthase onto a support, and then we have a test tube of compounds. They can be hundreds of thousands or millions or even more than millions of compounds, each of them synthesized with also a different DNA tag. Yeah, and the idea is that you have some kind of a binding assay where you fish out those small molecules shown with the spheres that bind to the shape of the GYS. And it allows you to have a barcode so that you can basically almost look like you have a scanner to allow you to identify which exact compound out of the millions you have in the test tube that really act on GYS. Yeah, so this is called DEL in the field of industry drug discovery. We're really fortunate in Newcastle University in the UK and in a few academic institutions where this DEL platform can be implemented. Yeah. So usually involves what I just mentioned, a simple test tube with compounds, doing the screening assay using some kind of DNA sequencing as the scanner to find the barcode. And then with really quite big data of screening data, there can be machine learning, there can be computational analysis to identify what are those that bind, and then use some kind of machine learning to feedback and predict other ones that could bind, or continue to add more compounds to the tube to repeat and to get better compounds. Yeah, so it's really from that test tube like that we do the screening experiment. Yeah. I'm not going to go in details here, but you might wonder what type of compounds are these. DNA encoder library are usually built upon building blocks. Yeah, they're sort of little synthons, and they usually are building blocks of 3 of them added together in sequence, or in certain cases in branch. Each of the 3 building blocks will have its own little piece of DNA barcode, and therefore the different permutations of these building blocks together and with their DNA sequence will then give you a really unique barcode like what you see here. Yeah, and that really provide the permutations of building up a large library based on a starting point of a few hundreds of building blocks. So we delighted that we have that set up, and we are now really intensely doing that DEL screening. And I say intensely because, as I say, this is an iterative process that we can keep adding more compounds, and we can keep generating more permutations. Yeah. So I'm very delighted to share with you that tomorrow the graduate student, PhD student in my lab, Brianna, is going to present the talk. And I really am very grateful to the APBD-RF Foundation for having a section for early career researchers or next generation scientists. I think that's very important for them to have the platform and to be able to partake in this community. Yeah. So that's one approach we're doing, and we call that high throughput screening approach. Yeah. The other two approaches that we are doing and we will be doing focus on, in this case, guided design of molecules based on known native ligands. Yeah. So I show here on the right-hand side the 3D shape of glycogen synthase that we know based on our own work and other people's work in experiments. I show ligands that we know that either target the active site, for example, the NLOC23 compound from Thomas Hurley's lab and late Peter Roche lab. Yeah. We also know allosteric activator ligands in another site known as the glucose-6-phosphate. Yeah. And we are also aware of other ligands in literature that also act as activators. Yeah. So our approach here is really use these known ligands and potentially known binding mode, and then design and modify them to build molecules. Yeah. So that's a guided approach, focused strategies. And we know that in this case, we are targeting 2 of the sites that we know. Contrary to the first approach that I talked in DEL, we do not know where it binds, because we're just opening the protein shape for molecules to bind. Yeah, so those are 2 different approaches. And very, very lastly, I always enjoy talking to Birch, and we often mention the word PROTAC. This is an approach that Birch invited me and others invite us to think about, and particularly when we try to develop small molecules that itself may not reach the necessary potency or affinity, yeah, or it may not reach the right site to do so. And in those cases, those silent molecules, as we talked about it, could have a potential utility as being used to fuse to known bait that target proteins to degradation mediated by ubiquitination. So this is called PROTAC approach, targeted degradation by ubiquitination, usually involving a binder or a molecule targeting the protein of interest, in this case GYS, and then with a chemical linkage fused to a known moiety that can engage the ubiquitin degradation machinery called the E3 ligase. There are, I think that a lot of people will know here that there are molecules being developed for PROTAC for various types of diseases. They're very well developed chemical entities for the blue one. And for the pink one, like what I have been saying, from approach 1 and approach 2 that we're doing, if they pursue to be potent molecules themselves, that's great. Yeah. If not, I think my thinking or our thinking here is those molecules that specifically bind to GYS could then be used as a PROTAC design. Yeah. And PROTAC mediated ligands in other diseases have been in clinical studies. Yeah, and very close to trials. Very close to approval. Yeah. So I think that's what I have as slides for today. Yeah. Just to reinforce the idea that we're very grateful for APBD-RF funding to kickstart and to allow us to really continue the DEL screening work. Yeah. And we continuing different approaches of hit discovery, like what I mentioned in terms of guided design and PROTAC design. And our usual workhorse of recombinant work of binding assay, kinetic assay, and structural studies are all in place. Yeah, and if there are other ways we can use these tools and share these tools for your characterization, we're very happy to collaborate. Yep. So I'm just going to end by putting the slides on and thank particularly those on photos who have been working on this work, and thank APBDRF and $1 Million Bike Ride for their support. Thanks very much.
Thank you, Dr. Yu. That was an awesome presentation. Now I'm going to open up the floor for question, and I'm happy to see that there were a lot of questions given in the Q&A function of the Zoom call. And maybe I can just bring the attention first to Dr. Gentry, and I'm just going to ask you first, you know, a lot of work has been done in Lafora disease. Now we're talking about APBD. Can you maybe just summarize how you see differences and alignments between what worked maybe in LD and Lafora disease mice? Why is it also supposed to work in APBD mice? What are the successes? What are the caveats of making this transition?
Yeah, I mean, I think the two big ones, great question, are the blood-brain barrier. Where are the aggregates, both in cell type, in tissues, and in subcellular location? So I think for all of these GSDs that we're talking about, and today the APBD, where are they at? And how do we get, in my case, an enzyme into those locations? We know that traditionally myozyme functions for Pompe disease. That's going into the lysosome. Presumably that would not work for APBD. We've not done that experiment, but we have tested it for Lafora's disease mice, which similarly has a glycogen aggregate in the cytoplasm, and it does not work there. So, you know, I like trying to have to put these diseases into common baskets and trying to answer the question, for that basket, what do we have available to target the aggregate? So yeah, I think location in terms of organ, cell type, subcellular location of the aggregate, if you're talking about an enzyme. Now, if you're talking about substrate reduction therapy, similar to Dr. Yu or Menashe, you know, I'll let them expand on that. But in that case, you don't care so much about subcellular location because you're turning the faucet down, right? You're trying to decrease synthesis. So those therapies don't have that piece of the challenge. Happy to hear their comments on that too. That is useful.
Yes, Dr. Menashe, anything you want to add?
I mean, yes, we don't have that challenge, but back to your original question, we think that getting rid of the polyglucose antibodies or preventing their formation will be therapeutic, you know, for both diseases. So there's many common approaches here, with the exception of gene therapy, which by definition is unique. You know, the other approaches should work equally for APBD. Obviously, there's a very important place for gene therapy because you cannot get more specific than that. That's a very important thing to keep on developing. So,
yes. All right. So then, thank you for the explanations. What about administration route? How do different treatments require different administration routes? We've heard about ASOs, gene therapy, small molecules, and enzyme therapies. What is the treatment regimen that you kind of envision? I think that this might be interesting also for the patient community.
so for the ASO, which is the closest to the patients because it's about to go in actual human beings in the sister disease, the treatment is by lumbar puncture every 3 months for the life of the patient. For other treatments, small molecules usually are given by mouth 1 or more times a day. The gene therapy is usually one and done. You do it, the gene goes and replaces and is functioning for the rest of the patient's life. And for the enzyme therapies, it depends, how it ends up being, but it's a short period of infusion, whether it ends up being into the brain or IV. I'm sure Dr. Gentry is thinking a lot about these things, but there again, it's a matter of getting rid of the polyglucose antibodies that have accumulated for 50, 60 years in APBD. And hopefully it's one and done, because once they're gone, it'll need another 60 years to accumulate enough to give symptoms, which obviously becomes kind of not so relevant.
Thank you. So I think the blood-brain barrier was mentioned a couple of times by Dr. Gentry, but not by Dr. Yu. How are you envisioning to have your compounds cross the blood-brain barrier?
Thanks, Felix, for the question. I think Dr. Orkalon also asked a similar question about blood-brain barrier in terms of compounds. Yeah. So obviously, the choice of pursuing small molecule therapy is one of which is the potential to be brain penetrant, yeah, or CNS penetrant. There are increasing ways to predict molecules of predictors of CNS penetration and of ability for permeability. So a lot of library designs these days, yeah, are steered towards those. Yeah. But of course, there's also the challenge that when small molecules become optimized and become better to target specifically for the enzyme, usually end up getting those small molecules too big that create problems in CNS. Yeah. So my very vague answer is yes. This is the key reason we want to pursue small molecules, but along the developmental path, it is something we often have to fail upon before we find better molecules. Yeah.
I see. Now, Dr. Choi and her gene therapy kind of solved the blood-brain barrier issue by using a serotype AAV9 that is like able to cross the blood-brain barrier evidently. Now, can you explain, Dr. Choi, what your considerations were regarding the choice of the promoter? You used a neuron-specific promoter. Why did you do that? Is that something you're envisioning for like a clinical approach as well?
Yes, thank you for the question. So first of all, to penetrate the AAV to the PBDB, so that's why we chose AAV9. And first of all, because APBD patient present a lot of neurogenetic phenotypes. So first we thought targeting a neuron for the cure. So that's why we chose a SNAP-SYN1 promoter, just only for targeting the neuronal cells. So after AAV treatment, as you can see previously, we saw the early benefit with the AAV induction, human GBE, and it successfully prevent the polyglucose antibody in the brain for 3 months. But unfortunately, after 6 months, we saw the polyglucose antibody begin accumulation. So now we are thinking about the complexity about the disease progression, not only neuron involved. So our next goal is to, now we are studying about the natural history to exploring the which cell type is involved. So we are trying to targeting different cell type to prevent this progression of disease.
And do you think the choice of the promoter was responsible for the decline, or is it just that the polyglucose antibodies became more present in other cell types?
Our first interference is the promoter is the main cause.
Culprit.
Yes.
Yeah.
So now our next goal is switch different promoter to target other cell type.
Mhm. I get it. Okay, that's great. So then I think we're almost finished. I would like to give one interesting question the floor, and I would like to also encourage all the panelists to look at the Q&A section and maybe look through questions that were addressed directly to them, and maybe if I couldn't bring the question out here to write a written answer there so that interested people don't go home empty-handed. One question that I would like to bring out here is a question from Rob Schreier for Dr. Minassian. Your comment about the slow progression of the disease for clinical trials was very important. What unique and additional data can we collect on patients that will mitigate the slow progression and give more data points to compare before and after interventions? Would be interested in your thoughts. And great talk.
Yeah, thank you. Honestly, I'm not the best person to answer that. I'm a child neurologist now and deal with different types of conditions. Certainly, more biomarker data will help. We don't work in the adult world. How do Alzheimer's disease patient communities do clinical trials? Obviously, Alzheimer's disease is a much larger disease with lots of money, and you can do long studies. I just thought to invite the community to talk to people who work in adult diseases, certain forms of ALS, Huntington's, Parkinson's, and so on, to figure out the best ways to conduct clinical trials. Alternatively, if there's a drug that is making it, to the market, let's say for a severe pediatric disease, could it be used in adult patients off-label? I don't know, and we don't have the time to delve deep into this here, but I just found that I'm just frustrated, to be honest, that we have a drug that should work in APBD. We're getting it into the Lafora patients. What can we do to get it into the APBD patients? It's something the APBD community should try hard to kind of figure out.
Thank you.